Okay, let me now introduce Bob Young, while the screen is being prepared. So Bob Young was named Genome GBC Research Fellow in 2007 and is the Merck-Foss BC Discovery Chair in Pharmaceutical Genomics, Bioinformatics and Drug Discovery. So Bob's lab at SFU conducts research to undertake the design and synthesis of small molecule probes to aid in understanding disease processes and to discover novel targets and therapies for treatment. A lot of experience, Bob, with Merck and others, and we're really pleased to have you here.
Thanks, Bob.
Please welcome Bob.
So it's a pleasure to be here, and sort of after a bit of time associated with Genome BC to maybe give you an update. I thought I would try and update you on things going on in my lab. I thought I could sort of give you an overview of the projects. Actually, when I started to do that, I realized that you end up with a lot of little bits of nothing and not much of anything. So I'll do that. I'll give you a quick overview, but actually try and get into something real. So obviously, genetics and the genome has given a lot of opportunity for drug discovery, opening up new targets. But it also has ways of perhaps understanding old targets or old drugs, which is very useful potentially. And of course, there are many techniques that have been developed to do genetic validations, which are useful. And so this is really a great opportunity for this chemistry or drug discovery in general. From an academic chemist point of view, obviously this is maybe a little more tough. When I worked at Merck, we had legions of biologists and molecular biologists, etc. to work with. I'm sort of in there on my own, and so it's a bit tougher to try and muster a full-blown drug discovery project, but we can work on old drugs, perhaps trying to understand how they work, trying to make probes, determine what the targets are if they're unknown. We can try and develop proof-of-concept molecules for new targets that are either proposed by ourselves or others. And we can try and develop targeting approaches to take known drugs and to make them better by targeting tissue in particular. And of course, to do all this, we need to probably collaborate in most cases, because I really don't do all that myself. We have a variety of equipment available to us in our lab, including particularly a high-resolution mass spectrometer, which is thanks to Genome BC. And I must say, when I got that, or when we were offered that, they said, well, you know, the equipment you have, which we had ordered for our lab, if you have all that, what would you like on top? And so I thought a long time, and I thought, you know, a high-resolution mass spectrometer would be useful if I really had everything. It turned out we got that first before other equipment came in, and in fact, it's been not only useful, but invaluable. So I have to thank Genome BC for that. We have another mass spectrometer we got from Merck Frost and HPLC systems and so on. We got from CFI, other methodologies for fluorescence readers and so on. And of course, we do synthesis. And we can do, like I say, synthesis, analog synthesis. We also have a radiochemical lab here, so we can make labeled compounds, which can be very useful for either finding probes for targets or for tracking drugs in the body. We have developed protein labeling and purification technologies, and we can do pharmacokinetics, drug metabolism, affinity probes, and pro-drug design, and other such technologies. So we really are well equipped to help out in a variety of approaches to drug design. And my current research projects involve a number, probably too many, considering we're only about 6 or 7 people. But we have a project to develop molecular probes to identify a novel target, hopefully novel target, for some anomalous inhibitors of glucotriene biosynthesis. These are inhibitors which work very well in cells. They don't seem to hit the known targets. So the question is there another target there? A major project we're involved with is in trying to develop inhibitors of macroautophagy to look at the role of autophagy in cancer and other diseases. It's a big collaboration with UBC, UVic, the Cancer Institute, and for which we got a very nice grant recently. We've been looking at bone-targeting prodrugs for delivering anabolic prostaglandins to the bone and potentially as a therapy for osteoporosis. And then we've been recently developing selective and specific pyruvate kinase inhibitors as potential antibacterial agents. I like that because it's totally against all pharmaceutical rationale to hit a target that was present in mammalian circulating cells. And so, in fact, this seems to work quite well. And then we're looking at actually a new project just starting out to modulate the androgen receptor, which is highly relevant because in the last talk, that's really only just started. So I can't get into all that, but I will try and discuss a couple of targets if I can, if I have time. And in particular, the most interesting at the moment is this study of macrophagy and its potential role in disease. And of course, so many of you may know, but the autophagy or macroautophagy is a process that takes place in most cells where there's an engulfment of cell contents in a so-called autophagosome, which is then merged with a lysosome, and those contents get degraded. It's a way that the cells recycle defective proteins, organelles, bits and pieces that you don't want to have around in the cell. And it's known there are a variety of gene products that are involved in this autophagy process, and in particular an enzyme known as ATG4, which cleaves pro-LC3, which is a key protein involved in autophagy. And that protein then is lipidated. It stimulates the formation of those autophagosomes. And then when those autophagosomes are formed, it has to be delipidated in order to facilitate the merger with the lysosome. And it turns out the ATG4 does both the pro to LC3 cleavage and also the delipidation. So we felt, and others have felt, that this might be a good target as a way of inhibiting autophagy. Autophagy is considered to be a normal housekeeping process, and obviously you say, why do you want to do that? You're going to kill yourself. But it's also upregulated in response to stress, such as starvation and anaerobic conditions. And it's felt, or it is upregulated in cancer cells, particularly rapid-growing tumors, or those that are treated with anti-cancer drugs. So an inhibitor of autophagy might synergize with anti-cancer treatments, and therefore be specific for rapid-growing tumors or in combination with anti-tumor drugs. And unfortunately, there are no selective inhibitors of autophagy available, so it's very hard to test this concept. There are many efforts going on to do that genetically, and that's part of our project as well. But ATG4B cleaving this key protein LC3 allows, is a target we thought would be useful to see if we could control the process. And ATG4B is a cysteine protease enzyme. I have, in my past history, been involved in designing cysteine protease inhibitors, so I thought maybe that could be something we could do. This is a collaboration with Sharon Gorski, Steve Jones, Nurse Salvelli from the Cancer Institute, Julie Lum from Victoria, Helen Cote and Keith Humphries from GBC, and also in my case, Alan Edwards from the Structural Genomics Consortium in Toronto, who has been very kind to provide us the protein and is now doing crystallography with us. And also Michelle Robert, Tom Pfeiffer, and Nicola Thompson at CDRD, who are involved in developing a screen and running a screen. And of course, I mentioned we have this grant, which we very happily got from CHR. Several X-ray structures are available for this protein, and it does have homologs, actually there's 4 of them. A, B, both have X-rays available. And they have also the bound protein with the substrate bound to it is also available. So this is very obviously useful if you're trying to design an inhibitor. Unfortunately, there's large conformational changes when the protein binds its substrate. And it's also auto-inhibitory. There's a loop which blocks the active site, which appears to somehow open up when it binds to the substrate. And there has been suggestions that it would be very difficult to get a small molecule inhibitor because of this self-inhibitory blockade. And so the question is, could we find an inhibitor? Could we design one? And really, it was an open question. For this reason, also, it was very tough to get small peptides that would be cleaved by this enzyme. So people tried to get assays, and they were unable to do so. So we had a problem. We had a good enzyme. We had the substrate, but we didn't really have an assay. We wanted to get kinetic assays so we could at least characterize our inhibition. We needed those assays somehow. We wanted to develop functional assays of cells. We wanted to synthesize inhibitors. We thought, first of all, we would try and make irreversible thiol-trapping inhibitors. At least they might have a better kinetic advantage to inhibit a tough enzyme like this. We want to make, of course, mechanism-based reversible inhibitors, which would then hopefully be better behaved in vivo or in cells. And of course, we'd like to get the crystal structures of those inhibitors bound to the enzyme, and also perhaps an homologous enzyme to see if we can get selectivity. And then we were going to try and optimize these hits or leads into really useful, hopefully in vivo useful compounds. The mass spectrometer, which was kindly available from Genome BC, it turned out to be invaluable because we were able to use an LC-MS assay. We could in fact see in the LC-MS the LC3 mass very easily. We had purified enzyme and purified substrate. And when you mix the two together, basically A and B, incubate them, you see the cleavage, and you see the loss of 559, which is the pieces cleaved off. And you can actually follow this peak going down to peak going up. And we could use that actually as an assay in real time. In doing that, we could show progression of cleavage, and we could show that an obvious viable shot like NF-molamide could in fact inhibit the enzyme, and it did it in a time-dependent fashion here. This is after 15 minutes, after 1 hour of pre-incubation. And it was obviously reacting with the enzyme and blocking the activity. We then went on to try and find a fluorescent peptide as a substrate. We didn't really believe the literature, so we did scans of peptide libraries. We designed fluorescent peptides, and we did find, in fact, a number of peptides that were cleaved by the enzyme, although very slowly over hours and even days, depending on how much enzyme we used. But we could see progression, and it was actually relatively useful, but it did require a lot of enzyme to get this assay to work. We could show a nice Michaelis-Menten kinetics, get a K in around 400 micromolar, which was not particularly good, but at least it was behaving itself. And we also made a variety of substrate, at least inhibitors, based on that substrate. And these were halomethyl ketones, where the glycine component was replaced by a halomethyl ketone, either fluoro or chloro. It's known that chloromethyl ketones are very reactive, fluoromethyl ketones are less reactive. So we weren't quite sure what would happen. We can show, for instance, the chloromethyl ketone inhibited irreversibly the enzyme, and it had a KIR of 8 micromolar. This is with a pre-incubation of about 30 minutes. Oops, that's not supposed to be there. This one. Eaten a few of my slides. Should do that. However, well, actually, I think it comes in later. So we then went on to try and develop a fluorescent assay, but we decided that perhaps if we could try and make a fluorescent LC3, we could actually have the natural substrate or something like the natural substrate, which would hopefully be converted at a more rapid rate, but it could also be followed by fluorescence, and it would derive a useful assay. And we decided to append 2 fluorescent proteins on either end of the LC3. Looking at the options, we decided on emerald-green fluorescent protein and yellow fluorescent protein because of their emission and their excitation emission spectra overlap. So we expected we'd be able to fret between these 2 fluorescent proteins. And it was known from the X-ray that the actual 2 ends of the LC3 were relatively close to each other. And it was also known that we could make a GFP LC3, which would be cleaved by the enzyme. It was also known that a GST fusion on the C-terminus was also cleaved. So it seemed that the enzyme didn't mind too much what was on either end. So we tried to make this construct.